Okay, let's dive right into this truly fascinating new study that completely reimagines how we measure and honestly, how we map human brain activity over time. Welcome to The Explainer. Today we're unpacking a groundbreaking piece of research surrounding something they're calling neurobytes. If you have ever wondered how the chaotic, constantly firing neurons inside our heads actually organize themselves into coherent, crystal clear thoughts, well, you're definitely in the right place.
For decades, scientists have been up against a massive challenge. They've been searching for a reliable neural biomarker, basically a clear signal, like a fingerprint for the brain, that stays totally consistent whether you're fast asleep, solving a really tough math problem, or even battling a neurological disease. But here's the catch. Brain data is incredibly noisy. It's highly specific to whatever task you're doing right that second or whatever machine is recording it. Finding a universal feature that remains reproducible across completely different experiments, recording systems, and clinical populations, it's been exactly like looking for a tiny needle in a massive, ever-changing haystack.
So, Part 1, the brain's hidden rhythm and how we measure its complexity. To really grasp this quest, we have to look at how we've traditionally measured these complex brain signals. Historically, using standard EEG analysis, we've treated the brain almost like an old radio broadcasting static frequencies. We measure spectral power, oscillatory bands, or specific event-related potentials. But the ECPJ framework, which stands for the Electrical, Chemical, Electrical Pierjanti framework, completely flips the script. It moves us away from looking at static anatomical structures and instead views cognition as a flowing network. It's a continuous succession of transient integration states. Essentially, the brain is constantly shifting and flowing, not just sitting still. Which brings us to Section 2, Meet the NeuroByte, our dynamic timing event. This is really the core of the proposed solution. A NeuroByte is officially defined as a temporally bounded dynamic timing event. In plain English, it's characterized by a quick burst of increased neural coordination and information integration. Think of a NeuroByte like a digital data packet in a computer network. It emerges when distributed neural populations sync up, it stabilizes for just a fraction of a second to integrate information, and then boom, it collapses so your brain can move right on to the very next thought. It is literally the dynamic unit through which information is transmitted across your neural systems. Now for Section 3, the ultimate reproducibility test, via a cross-cohort pipeline. Because, right, it's one thing to have a fantastic theory, but it's another entirely to prove it actually works in the real world. Can we reliably extract these dynamic neurobytes everywhere? Reproducibility is the absolute fundamental requirement for any biomarker. So to test this, the researchers built a highly rigorous, standardized 6-step extraction pipeline. It goes like this. First, a bandpass filter from 1 to 30 Hz to isolate the right frequencies. Second, a Hilbert transform. Third, z-score normalization. Fourth, they set a really strict threshold at 1.8 standard deviations above the baseline. Fifth, they constrain the event duration to between 30 and 500 milliseconds. And finally, feature extraction. This rigorous pipeline guarantees every single piece of data is treated exactly the same way, no matter what. They then unleashed this exact same pipeline on an incredibly diverse set of independent cohorts. And when I say diverse, I mean they really threw everything at it. They looked at resting state EEGs, people doing mental arithmetic, visual and auditory tasks, and even sleep and anesthesia datasets. But it didn't stop there. They tested data from healthy aging cohorts, patients with Alzheimer's disease, Parkinson's, ALS, multiple sclerosis, and get this, even healthy people learning new surgical skills. The goal here wasn't to diagnose a specific disease. They just wanted to see if the pipeline could consistently pull out these neurobytes from vastly different states of human consciousness. Moving into Section 4, Universal Brain Metrics and the Cross-Cohort Results. So how did this massive experiment actually play out? The results were absolutely striking. The pipeline successfully generated timing metrics across datasets that had markedly different experimental designs and recording objectives. The researchers proved that neurobytes are consistently detectable across all these independent datasets, whether a subject was wide awake doing complex math, deep in sleep, or suffering from severe neurodegeneration, the candidate neurobyte events were there. This strongly suggests that these metrics capture a fundamental, general property of neural timing organization. It's not just a fluke or some artifact specific to one particular lab's dataset. To make sense of all this, the pipeline generated a standardized payload of timing data for every single recording. They computed very specific metrics, event count, event density, mean event duration, and the time between those events, which they call the inter-event interval. or IEI, along with its variations. They even calculated an overall stability index. What this does is provide a universal mathematical vocabulary for how the brain integrates information. Suddenly, we have a completely standardized way to compare the neural timing of someone in a deep, dreamless sleep directly against someone learning to perform complex surgery. It's wild.
And finally, Section 5, the timing, fingerprint library, and mapping conditions. The implications of this successful computational test are, quite frankly, absolutely staggering. Now, it is important to note the study's limitations. This demonstrates computational reproducibility, sure, but it doesn't quite establish biological causality just yet. Independent replication and multimodal imaging are definitely still needed by external labs. However, the ultimate vision here is brilliantly captured by the researchers themselves. They state, rather than relying on a single biomarker, this approach seeks to map how different conditions alter neural timing architecture. Just imagine a comprehensive timing fingerprint library. In the future, every physiological state, whether it's deep focus, fatigue, or a pathology like Alzheimer's, could be identified not by a static image, but by its distinct dynamic distribution of neurobytes.
This research represents a massive leap forward in viewing cognition not as some rigid, static structure, but as a beautifully choreographed dance of time. As we finally begin to map these dynamic units of human cognition, we really have to ask ourselves, what unseen rhythms are dictating your thoughts right now? What other secrets is our brain's timing architecture just waiting to reveal? Thank you so much for joining me for this explainer, and I hope this gives you a completely new perspective on the flowing network inside your own mind.